Hello, my name is Nafeesa. My metric number is E21430695. Today, I will present about determination of microbes in deer captivity. So, why I interest with this research? This is because of I have high interest to study on microbes. The introduction of microbes in deer captivity is a critical issue that poses significant risk to the health of captive deer population. Deer captivity is a common practice in many parts of the world where deer are kept in enclosed areas for various purposes such as breeding, hunting, or entertainment. Microbes, including bacteria, viruses, fungi, and protozoa, can cause various diseases in deer. Several studies have reported the presence of various microbes in deer captivity worldwide. The presence of these microbes can lead to significant economic losses due to animal mortality, reduced productivity, and the cost of implementing control measures. It is necessary to prevent the introduction of new infectious agents and to control the spread of existing ones in captive deer populations. Effective management strategies such as quarantine, vaccination, and improved sanitation can help prevent the spread of infectious diseases. Additionally, regular monitoring and testing of deer populations can help identify any potential health risks and allow for timely implementation of control measures. So this is the objective of this research, which is environmental management. This is to provide information about the overall environmental health and sanitation practices in deer captivity. This is three problem statements included, which is disease surveillance, environmental health and sanitation, and lastly is conservation and management. Firstly, disease surveillance. It is difficult to put in place efficient disease surveillance programs without a good understanding of the microbial ecosystems in captivity deer. Second is environmental health and sanitation. The environment's microbiome has a profound impact on the health and well-being of confined deer. Lastly is conservation and management. Deer population kept in captivity frequently serve as genetic diversity and conservation efforts reservoirs. Next, this is significance of study, which is effective wildlife health management. Second, certain bacteria can be harmful to the health of captive deer populations. And lastly, it will further our knowledge of host microbe interactions, microbial ecology, and wildlife microbiology. So deer are commonly kept in confined spaces for a variety of reasons, including conservation, research, and breeding. This practice is referred to as deer captivity. In order to manage and study these animals in regulated environments, captive deer populations are established in wildlife reserves, zoos, research facilities, and game farms. To identify microbial population in captive deer, a variety of techniques are used, including traditional microbiological, DNA sequencing methods, metagenomics, and next generation sequencing (NGS). Factors can influence microbial communities in the area of deer captivity. First is environmental microbiota, second is wildlife interactions, and lastly is human activities. How infectious diseases can have a significant impact on deer health, population dynamics, and the general management of captive facilities can affect population of captive deer. First is close contact and overcrowding. Second, captive deer frequently share water sources and feeding stations. Third, captive deer may experience long-term stress, which may weaken their immune systems. So, what are effective disease management strategies to reduce the risk of disease transmission among captive deer population? Firstly is biosecurity measures. Second, routine disease surveillance is essential. Thirdly, quarantine protocols should be in place for newly introduced individuals. And lastly is the captive environment must be managed properly. This is methodology, which is firstly sample collection, Secondly, microbial analysis. Thirdly, data interpretation. And lastly is interpretation and reporting. In the next slide, it will be shown my methodology poster. This is the expected outcome of this research. Firstly, disease risk and pathogen detection. Secondly, environmental impact and management. And lastly, scientific understanding and future research. For the budget, to travel to the fieldwork, the transportation is 30 ringgit Malaysia, and for food and meals is 25 ringgit Malaysia. The location of fieldwork is not far, so the transportation is cheap. So the contingency funds is between 50 ringgit to 100 ringgit Malaysia. So for the three last slides, there is the Gantt chart of the activities that have been done through the month of April to June. For the milestones, it shows the date of starting date doing the e-portfolio and the end date finishing it.